Good evening, Akansha.
Hello.
Hello, Akansha, can you hear me, dear?
Yes, ma'am. The voice is very low.
Now, can you hear me?
Yes, yes.
Yeah. Good evening, dear.
Good evening, ma'am. Should I switch on video?
Yeah, you can. You can. Yeah,
Okay.
please. How are you, Akansha? Going good?
I'm fine. Yeah.
Okay. Chalo, let's begin with our lecture. All right,
Okay.
dear. Okay, dear. So the thing is, already started, we are studying with the reproduction in higher and lower plants, right? We have
Yes.
completed with our patch, right? We have completed with sexual reproduction we are going on, right? In
Yes.
asexual reproduction, dear, we have studied with fragmentation, budding, spore formation.
Performance.
Yes, in vegetative reproduction, we are continuing with cutting, grafting, right? We have studied sexual
Yes.
reproduction. We have saw all the sexual floral whorls. Okay. Then
Yes.
even we have studied with a structure of anther. We have dealt with
Yes.
PS of anther, structure of microspore, which is related pollen grain. Then we have studied with development of male gamete. Then in a
Yes.
last lecture, we studied with structure of anatropous ovule. Right.
Yes.
So let us revise once. Okay, before starting with our new patch, let us revise once the structure of anatropous ovule. Can you see, Akansha?
Yes, ma'am.
Yeah. Structure of anatropous ovule, dear. This is a diagram structure of anatropous ovule, right?
Yes.
And in this anatropous ovule, we saw the female reproductive whorl, right? The ovule means it
Yes.
is a female reproductive whorl. It is called as an carpel. This carpel consists of ovary, style, and stigma, right?
Yes.
Even we saw that anatropous ovule consists of two parts, that is funicle and body of ovule. In
Yes.
funicle, dear, we studied that it is what is a stalk. Always the stalk is variable in length. If the
Yes.
funicle consists of ovule, we call it as an funiculate ovule. And if
Yes.
funicle doesn't consist of ovule, it is called as an sessile ovule.
Sessile.
Yes. Then we studied what is the function of and the funicle. The function of funicle is that to connect the body of the ovule to the placenta. Even we saw the body of ovule, it consists of an integument, micropyle, and chalaza. We saw that integument, it is two types, that is outer integument and inner integument, right?
Yes.
And what is the meaning of an integument? Integument, it is nothing but which is covering externally, and it is consisting one or two more layers of nucellus, right? After that,
Yes.
saw that integument, it is what? It is if there is a presence of one integument, it is called as a eutecmic ovule. If it consists of an two integument, it is called
Due technique.
yes, butechmic ovule. Then the function of the integument, we saw, Akanksha, that it gives protection to the nucellus. Micropyle, micropyle, it is nothing but it is a what? It is giving, it is surrounded by an integument, right? Completely surrounded, and it is in small opening. And what is the function of micropyle? Micropyle act as in what? It is a passage for pollen tube during the
Pollen tube.
yes fertilization purpose, right?
Yes.
Then we studied about chalaza. Chalaza, what? Chalaza, it is a base of the nucellus. Into the anatropous ovule, we have saw on the upper side there is a presence of an what? Chalaza, and at a
Yes.
lower side, if we will see the
Micropyle.
yes, there is a presence of an lower side micropyle, and at the upper side there is an chalazal pole, which is called as an chalaza, right? Then even
Yes.
we have so moved towards what? We have moved towards the nucellus. Nucellus, it is a major part of the ovule and definitely
Yes.
made up of the main parent tissues, which is called as in diploid parenchymatic tissue. Even we saw that there are two ends, one end which is opening towards the chalaza, it is called as in chalazal end, and the one end which is consisting, which is having a small opening, it is called as a micropyle, which is called as a micropylar end. What is the function of nucellus? On maturity, nucellus developed into a embryo sac.
Embryo sac.
Yes. Then we studied about the embryo sac, which is also called as a female gametophyte. Now we everybody know the embryo sac. When the nucellus get mature itself, it is called as an embryo sac. It is present towards where it is present towards the micropylar end. What is the shape? The shape is oval and haploid, right? And therefore our
Yes.
female gametophyte is also called as an embryo sac. It consists of an what? It consists of an seven cells and eight nuclei. So we call it
Yes.
as in seven celled and eight nucleated structure.
Nucleated structure.
You know, Akansha, in the last lecture I have made you study that how we counted, how we have dealt with
Yes.
that. Yes, it is why it is called as an seven cell and eight nucleated structure, right? Then after
Yes.
it, what does it consist of? The female gametophyte consists of egg apparatus, secondary nucleus, and antipodal cells. Now, in egg apparatus, we studied about micropylar end. Then we studied about the three cells, which is the middle one. It is what? Middle one. It is called as an egg cell.
Eggshell.
Yes, and laterally, the egg cell is surrounded by your two cells, which is called as in synergids.
Synergids.
Yes. Now, during function, what is a function? During fertilization, this egg cells fuses with one of the male gametes. How many male gametes are present, Akansha?
Two.
Yes, the two male gametes are present. So the first egg cell have been what fused with first male gamete to form a diploid zygote. And what is a
Yes.
synergid? Synergids, it is a cell which is playing a role during supportive role for the fertilization, and even it provide a nourishment to these cells. Secondary
Yes.
nucleus, you also know, I also know that what the
Yes.
secondary nucleus is present into where? Into the center within the embryo sac, right? Then
Yes.
we move towards that. It consists of an what? It consists of an two polar nuclei which are present in a center, and therefore they all are the diploid cells. Function. What is the function? During fertilization, the second male gamete which is present, it fuses with a secondary nucleus to form a triploid primary endosperm
Endosperm
nucleus,
nucleus.
right? Therefore, it
Yes.
is the short form, it is called as an TPEN. So overall, we have saw there is a presence of in two male gametes. Even we have saw when we have studied about the when we have studied about the what we have studied about the male gametophyte, there was a presence of in two male gametes, right? And one
Yes.
male gamete got fused, and it have formed what? It have formed a diploid
Zygote.
zygote. Yes.
Yes.
And one male gamete, it has got fused and it has formed secondary nucleus. And what does it form? It formed a triploid primary
Primary
endosperm,
endosperm
right?
nucleus.
Yes.
Yes.
Then we saw antipodal cells. Antipodal cells are present within female gametophyte at the upper side, right? And
Yes.
antipodal cells are three in numbers, right? That's why it has been said it consists of an three cells. Where is it present? It is present near the chalazal end, where we called as in chalazal pole also. What is the
Yes.
function? They are the accessory cells. Therefore, overall, if you will see into an embryo sac, there are 80 cells are present in embryo sac, and they all are monosporic. And therefore, they are present inside, hence we called it as an endosporic. And how are they? They are seven celled and eight nucleated set. And whenever the cell is seven celled and eight nucleated set, we called it as an polygonum type of cell. So the embryo
Yes.
sac is polygonum type, right, dear? In the last lecture, we
Yes.
have saw this. Even we studied this gametophyte. That what we have
Yes.
saw this gametophyte, female gametophyte, that how this megaspore went under a fertilization process, how many megaspores got degenerated, and then how in what way the megaspore was functional. Even we saw how many embryo sac, two nucleated embryo sac was present. Later on, it became four nucleated, then eight. All this has carried through via the cell division called as in meiosis. Right, Akansha?
Mitosis also happen, na?
Yes, mitosis also happened, but where the mitosis have happened? Can you tell me? During the megasporogenesis, isn't it?
There meiosis happen, na?
See, yeah, there the meiosis happened, right? The
Yes.
thing is that what there the meiosis have happened. The single cell which was diploid, the tetrad formation, okay, and
Yes.
then there is a degeneration. And yes, as said by you, it was right. But actually, what kind of and whether it was more mitosis or meiosis, Akansha, can you tell me?
Mitosis.
Is it mitosis?
Yes.
Okay. See, the functional megaspore form two cells, and from two cells, two nucleated, it become four. Now, can you tell me it is mitosis or meiosis? From
A
two, it
meiosis.
yes, it is meiosis. Okay. So if
Yeah.
anybody asks you, right? If anybody asks you or in neat question, if we have a question that what in an embryo sac in female gametophyte in floral reproduction, which of the majorly cell division have been taken place? Dear, we have to write it down meiosis, right? Because only in a functional
Okay.
megaspore there was a one cell. They it carried out
Yes.
mitosis, and from first nuclear division, the two cells was formed with the help of a mitosis, right? But later on, what
Okay.
happened? This mitosis cells, for formation of four cells, they went under meiosis, and with the help of a
Yes.
meiosis, how many cells came over here, right? Three nuclear division, third
8.
partition, yes, eight. बराबर है बराबर यहाँ पे सिर्फ दो
हाँ.
बढ़ा क्या आकांक्षा यहाँ से सेकेंड न्यूक्लियर डिवीजन से जब हम थर्ड न्यूक्लियर डिवीजन करने लगे तो ये फोर से सिर्फ दो बढ़ा क्या ओनली टू हैव इंक्रीज नो फोर डबल हैव गॉट बराबर है कि नहीं चार का डबल
हाँ.
हुआ यहाँ पर है ना अगर माइटोसिस
हाँ.
होता तो बेटा सिर्फ दो बढ़ता यहाँ पर है ना तो यहाँ
हाँ.
पर सिर्फ दो बढ़ा के ओनली टू हैव गॉट इंक्रीज नो अपर फोर
No.
एंड लोअर फोर बराबर है कि नहीं right?
हाँ.
So why it has been happened? It has been happened due to the meiosis activity, meiosis cell division, right? Then we went for a
Yes.
development of a female gamete, barabar? And this development of the
हाँ.
female gametophyte, which is called as an embryo sac, we saw that from here, from the upper side, where it was a four cells, that is an eight nucleated embryo sac, barabar? Upper four
हाँ.
and lower four. From upper, one cell came into a middle barabar into a center, and from lower it came to a middle, and therefore into a center we got a polar nuclei. So therefore there is a shifting of a nuclei has been carried out, right, during a female gametophyte. Okay,
Yes.
and this which is present, the upper side it is an antipodal, and the lower side it is what, synergids surrounding
Synergids and egg.
egg cells. Okay, and therefore this is a formation of an female gametophyte we have done. Even we saw
Yes.
with the embryo sac, right? This was a whole diagram of
Yes.
an embryo sac which we have saw. Okay, these are the two central cells which are the polar nuclear cell. Near the chelazal
Yes.
end we have a three antipodal cells. Micropilar end we have a three cells. In middle we have a egg cell, and egg cell is laterally surrounded by a synergid cells, right? Even we saw
Yes.
about what is the meaning of an archisporium, what is the meaning of a megaspore mother cells, megaspore
Yes.
genesis, right, dear? Then we came towards a double fertilization. In double fertilization, we saw that this is a diagram of a double fertilization. How these pollen grains have been germinated on the stigma, and then what way this stigma got swell and the pollen tube have been out. Barabar? And this pollen tube,
Yes.
which we called it as in germ tube also, it has been passed on from style, and from the micropilar end it has been what it is doing, it is acting, doing the further process with the help of an embryo sac, right? So that's why how
Yes.
this whole germ spore, that is a pollen tube, have interacted with synergid, egg cell, even the polar nuclear cell, antipodal cell, isn't it? Therefore, what
Yes.
we have saw into a double fertilization, dear, we saw into a double fertilization. That double fertilization, it is nothing but how many? The two male gametes which are present, right? So one male gamete, we know that it gets fused with the egg cell to form a diploid zygote. And second male gamete fuses with a secondary nucleus to form a triploid primary endosperm nucleus. This process, it is called as in double fertilization,
Double fertilization.
right? Who was the
Yes.
scientist, Akansha, who have invented the double fertilization?
Navashin.
Navashin. And which were the first plants he have saw this experiment of double fertilization?
Liliaceous.
Liliaceous. The plant's name, Lilium and
And
Fritillaria.
fertility.
Yes. What is the process of double fertilization? We everybody know that there is a presence of an pollen tube which is called as in germ tube,
Germ
right?
tube.
Yes. That germ tube consisting of in two male gametes. We have saw in development of male gamete. Into a pollen tube, there is a presence of in two male gametes. Okay. And what happened? This male gametes are passes through from where it passes through the style to where they have to reach. They have to reach the ovary, and from the ovary they have to reach towards the ovule, that is our embryo sac, right? Now we
Yes.
everybody know, whenever the pollen tube, okay, enters into the ovule through micropyle region, it is called as in porogamy.
Orogamy.
Yes, and whenever as a growth of the pollen tube continues, what does it happen? It ruptures the wall of the what? It disturbs the wall of
Embryo.
the, yes, it disturbs this wall of the nucleus, right? And what happened?
Yes.
Finally, it reaches to whom? Finally, it reaches to the embryo
Synergy.
sac. Yes,
Yeah.
synergy cell, barabar. Okay, but which is a
Yes.
main cell? It is called as embryo sac.
Embryo sac.
एम्ब्रियो सैक में ही एम्ब्रियो सैक में ही ये सारे cells present है, right? So ultimate its
Yes.
main aim of the pollen tube is to reach towards the embryo
Embryo
sac.
sac.
Okay, so this is a diagram which we have saw. Even we have saw this of the diagram which is giving the description about the style, stigma, ovary, right? And on
Yes.
the stigma there is a presence of an pollen grain. All right. Then,
Yes.
Akansha, we saw what we saw, the pollen tube enter to the ovule. Okay. The embryo sac. One of the synergids, that is what the synergid has got fertilized. Therefore, the synergid, it does what? Before synergids, let me tell you that what the main intention is reach towards the embryo sac. But in embryo sac, one egg cell is present, and that egg cell is surrounded by whom? Laterally surrounded
Two
by the
synergids.
two synergids, right? And those synergids,
Yes.
the main aim is what? The synergid is what? It is giving development and nourishment to those egg cells, right? Now this pollen
Okay.
tube, what does it do? It absorbs the water, it absorbs the nourishment from whom? From synergids. And what happens?
Synergids.
Yes, and after absorbing the water and the nourishment,
It
the pollen tube gets
swell.
swell. Okay. Yes, after it getting
Yes.
swell, it get burst open, right? There is a one limitation for getting swelling. And once it swell, what happened? It get burst open. And what happened? It releases how many male gametes? Two male
Two.
gametes. Yes. Into whom? Into the embryo sac, right? The
Yes.
non-motile male gametes, because no, male gametes are not motile in condition, they are
Yes.
non-motile. So what they do, they are brought towards whom? Towards the female gamete. And what do? They are helped with the help of an whom? They are helped for the motile condition, for a movement. They are taking help of the pollen tube. So whenever the male gametes are non-motile and they are trying to reach towards a female gamete, and the help, the motile condition where it has been provided by pollen tube, it is called as an sphygmogamy. Okay, it is called as a
Yes.
sphygmogamy. We every
Pygmogamy.
Yes, and we everybody know that two male gametes are present. One male gamete fertilizes with egg cell to form a diploid zygote. When this first male gamete fertilizes with a fertilizes with a egg cell to form a diploid zygote, hence that fertilization is called as in first fertilization. and it is also called as in syngamy, right? Normally in common words also, Akanksha, always remember wherever there is in syngamy, it is called as in fertilization process. So fertilization is also called as in syngamy.
Singami.
Yes. Now the second male gamete, you also know, and even we also know that what it
Yes.
has been fused with a secondary nucleus to form a triploid primary endosperm nucleus,
Endosperm nucleus.
right? So this is a
Yes.
second fertilization process. Hence, it is forming a triploid primary endosperm. So we also called as a triploid
Triple
fusion.
fusion.
Yeah, triple fusion.
Yeah.
Okay. Now, both this male gamete have been participated into our fertilization process, and they have been fused at different places into the embryo sac. Yes, because first it has been fused with egg cell, and the secondary it has been fused with whom? It has been fused with a secondary nucleus. So both the male gametes which are present, they have been fused, they have been done a fertilization activity on a two different places. Okay, Akansha.
Yes.
So this was a part of a double fertilization which we have already studied in a last lecture. Now let us see why we are
Yes.
constantly studying with this significance of double fertilization, right? Now this significance of double fertilization, why this is so much importance we are giving it. Okay. Now this double fertilization is only seen into the seeds, flower and fruits bearing plants. Okay, which we called as in
Okay.
angiospermic plants. Am I right, Akansha?
Yes.
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 Now, in this double fertilization, we know that two male gametes are present. So, both the male gametes are what? Both the male gametes are doing the process and activity of the fertilization. So, both the male gametes are utilized. Am I right, Akansha?
Yes.
Yes. Whenever the fertilization takes place, it results in the formation of an diploid zygote. And whenever the diploid zygote is formed, it will form what? It will lead to the formation of our embryo and seeds which are present into the fruits. So what they want to say,
Okay.
hence just now I said you कि हम सिनगेमी को ही fertilization भी कहते हैं. तो जहाँ कहीं
हाँ.
पे भी fertilization होता है और फर्टिलाइजेशन होने के बाद जहाँ कहीं भी डिप्लॉयड जाइगोट बना, तो हमें एक चीज याद रखनी है कि वहां पर प्लांट्स में डिप्लॉयड जाइगोट बनेगा फर्टिलाइजेशन से, तो वहां पे पक्का है कि वहां पे एम्ब्रियो एंड सीड्स बनना ही बनना है, क्योंकि प्रॉपर
Okay.
फर्टिलाइजेशन सेक्सुअल फर्टिलाइजेशन कैरी हुआ है. Okay, so this
Okay.
fertilization which is happening, it stored the diploid condition of the plants, right? See, I have written here, syngamy restores the diploid condition of plants. Means what? Whenever you eat that fruit, for example, like mango, barabar, whenever we eat a mango plant, that mango plant is going to give the other plant which is diploid in condition, isn't it? So
Yes.
why it is giving a diploid condition? Why same activity have been carried out even in a mango plant? The reason behind that, the fertilization activity which is carried on, which is also called syngamy, it has been stored its memory data into the seeds, that whenever the people are
Okay.
going to see sow the seeds, whenever the people are going to eat those mango and throws the seed, right, and that seed is going to germinate into a new plant, it will be into a diploid condition. Understood, Akansha?
Yes.
ओके. इसीलिए कहा जाता है कि जब कभी भी फर्टिलाइजेशन प्रोसेस हुआ, तो डिप्लॉयड कंडीशन में ही दूसरा प्लांट्स मिलना है, right? Whenever there is a triple fusion which is carried
Okay.
out, okay, it will form a endosperm of seed. Soon we are going to see what is an endosperm. All right. But the thing is,
Okay.
whenever there is a triple fusion has been done, this triple fusion, it will be always resulting into a formation of an TPN. What is the full form of TPN, Akansha?
Triploid primary endosperm nucleus.
Very good. So this triploid primary endosperm nucleus, it is going to form what? It is going to form a endosperm of the seed. Understood, dear? What is an endosperm?
Yes.
Endosperm is that kind of a cell which is present into the seed, which is going to provide nourishment to the embryo. Okay, it will always give
Okay.
nourishment to the embryo. Due to this double fertilization activity into the angiospermic plants, into angiospermic seeds, the seeds are more viable, right? Seeds are more viable means the seed is having a longer lifespan. You can use
Okay.
the, yes, you can use these seeds in any kind of condition. Okay, why? Because this double fertilization activity is carried out. Okay, and due to this viability, due to the longer lifespan, what happened, the percentage of seed germination is very high. The seeds get germinated very high.
Okay.
Okay, means the rapid process is carried out into a seed germination. Understood? It doesn't take,
Yes.
see, always the seed is germinated depending upon its dormancy, right? Whenever the seed will lose its dormancy, what is going to happen? The seed is going to germinate. In the same way, right?
Okay.
In the same way, if I'll talk about now, for example, like you take it as in mango plant. Mango plant is very common, or rose plant, right? If today I am,
Yes.
what am I doing? I'm putting a seed of an rose plant. What it is going to happen after 1 or 2 weeks, I'm going to start getting a little bit, what little bit growth is going to see into that rose plants, right?
Yes.
I'll see the radicule, I'll see the plumule, isn't it? Why? Because the reason behind that is those seeds are angiospermic, right? The viability is more, double fertilization activity has been carried out, and the percentage of seed germination rate is also very high. Understood?
Okay.
That's why the seed
Yes.
has lose its dormancy. Means what? Dormancy means what? The seed has given permission to get germinate and to release its roots and stem. Barabar. But
Okay.
what is the meaning of radicle? The radicle is going to develop into, later on it is
Root.
going, yes, it is going to develop into root, and plumule later on it is going to
Shoot.
develop into a shoot, and from shoot it is going to develop into a stem,
Stem.
right, Akansha? So
Yes.
this is a process. Due to this, all such kind of an regions, the double fertilization activity has been carried out, and it has been seen that double fertilization activity is much more necessary in each and every plants. All
Okay.
right. So the first
Yes.
point of the double fertilization is it is only seen in which plants, Akansha?
Angiospermic.
Yes. In double fertilization, how many male gametes are utilized?
Two male gametes.
Yes, if the fertilization syngamy activity is carried out, what it is going to form? It is going to form a diploid zygote. This diploid zygote will form a embryo and seed.
Embryo and seeds.
Yeah, always the fertilization, okay, always the syngamy activity restores the diploid condition of plants. Okay,
Yes.
and they are going to show their own characters of diploid condition, of sexual condition, right? They are not going to
Yes.
say, chalo, this generation or this condition is going on, this time we are not going to function. Instead of diploid, we will play a haploid role. This is not going to be possible, right?
Okay.
And with the form of, and with this all this thing, this triploid fusion, right, when the second male gamete have been fertilized, there is activity which has been carried out, process which has been carried out, which is called as an triple fusion. And this triple fusion, it does the formation of TPN, which is triple, which is triploid primary
Primary
endosperm
endosperm nucleus.
nucleus, yes, and it is going to form a endosperm.
Endosperm of
Yes,
seed.
this endosperm always give nourishment to the embryo. Whenever this due to whenever into a plants, if you can see the double fertilization activity, angiospermic seeds are more viable. They are having their more lifespan, proper conditions, right? And therefore the percentage of seed germination is very fast and high. All right, Akansha, till here you have
Yes.
understood.
Yes.
Yes. Now let us move towards a further topic which we called as in pollen pistil interaction. Right? Now this pollen
Okay.
pistil interaction which is present, Akansha, if you will see this pollen pistil interaction, what you will see in this, you will see in this that, okay, there are the presence of an pollen grain, And this pollen grain which are present, they are deposited on the stigma. But what does this stigma do? The stigma just keeps its own pollen grain of its own plant, and rest all are degenerated on the stigma itself. Okay,
Okay.
so how the stigma and pollen grains interaction has been done? For example, like you are my student, I am your teacher, right? Now the
Yes.
interaction between we both is like what? Now continuously taking lecture. Now interaction between we both are comfortable with each other, right?
Yes.
You know me, I know you. In the same way, in a plants also, there should be a proper interaction. Means what? The proper
Okay.
identification of the pollen grain has been done by the stigma. Why? Because is it possible that on rose plant there are supposed to grow shoe flower? No,
No.
right? On shoe flower there will be a mango plant or mango fruit which is
No.
growing. No, right? This is something which is
Yes.
happening fishy into the nature, isn't it?
Yes.
So why, for such reasons only, what the style and stigma has done, we will identify our own pollen grain. Means if the shoe flower pollen grain has been brought by insect, the pollen grain will be identified by whom? By a stigma. If the pollen grain is of shoe flower, the style will keep it, the stigma will keep it, right? If some other plant pollen grain is there, it will get degenerated because,
Degenerated.
yes, because they are not going to give a proper nourishment, because it is not of ours plant, so we are not going to
Yes.
provide a nourishment to them. Now let us begin with pollen pistil interaction, right? Now this pollen pistil interaction, dear, this event means this all this activity is carried out on the stigma. Okay, means we
Yes.
everybody know that the pollen deposition has been done on the stigma, and after present on the stigma, what is going to happen? That pollen grain is going to release a pollen tube which is called as a germ tube, and with the help of an via what? Via the style process, right? Via the style pathway, that pollen tube is going to enter into a ovule, and after entering into a ovule, it is going to reach to the embryo sac, right? So till
Okay.
the ovule process, all this, even whenever the stigma has, whenever the pollen grains has been deposited on stigma, from there, the process starts, and hence it enters into the pollen tube till the ovule. It is called as an pollen pistil interaction. All right.
Okay.
That before starting pollen pistil interaction, I said you that what is a relationship? How is a comfortability and compatibility with the
Yes.
pollen grain and stigma? Understood? Because it is much more necessary to know each other. Then and then only the
Yes.
pollen grain is going to get deposited and the pollen tube is going to release, right? And even
Yes.
we everybody know that how this pollen grain has been allowing the entry of the pollen tube, because this pollen grain, it absorbs the sugary substances which is present on stigma, right? जितना कितना भी
Yes.
शुगरी substances है, वो हमारा पोलन ग्रेन क्या करता है? एब्जॉर्ब करता है.
Absorb करता है.
Yes, after absorbing, the germination of the pollen grain start on stigma, and the pollen
Yes.
grain gets swell, and after swelling of that pollen grain, the pollen tube has been released, isn't it?
Yes.
And that pollen tube passes via from where? Via from style, and it
Stigma.
reaches,
Style.
yes,
Style.
yes, it is style, and it reaches to the whom? It reaches to the our
Ovary.
ovule, that is the ovary. बराबर है कि नहीं? And
Yes.
from ovule it is going to reach to the embryo sac, right?
Yes.
Now during this pollination, it is not sure that stigma or pistil receives the pollen grain of same species. Absolutely right. That's why I said
How?
shoe flower is not going to say, Chalo, now pollen grain of the mango has been deposited on my flower. So today I will reproduce a mango on my tree or on my plant, right? Shoe flower will
Yes.
not say in this way. Or mango will not
Yes.
say, oh, my pollen grain has been flown to the
Shoe
shoe
flower.
flowers. Yes. So today I'll grow, or the shoe flower pollen grain has come to me. So I'll grow the shoe flower. It's not like that. Okay,
Yes.
there is what? It is not sure by a nature, that the nature is going to get a same kind of an pollen grain, right? Now it
Yes.
depends that how the dispersal, how the pollination activity has been carried out. All right, how the insects
Okay.
are doing the pollination, how the wind is doing the pollination, right? And how which
Yes.
of the pollen grains they are bringing on the stigma. Okay, now this pistil has ability to recognize the pollen grain, means the stigma itself have a capacity, what it is having a capacity to recognize the pollen grain. Understood? Without
Okay.
recognition, without identification, the stigma is not going to allow the pollen grain to come inside.
Yes.
Understood? Okay. If it is a right type, okay, if the pollen grain is of right type, stigma accept it and promote and pass the fertilization event. Okay, what it is going to do,
Okay.
if the pollen grain is of rose plant, okay, if the rose plant is
Yes.
present and the pollen grains have been deposited by what, by an insect, the pollination has been done by an insect on stigma of a
Yes.
rose plant, right? And the rose plant has received its own pollen grains that is of red color. Plants even see the color because we have
Oh,
roses of red, white, yellow,
yes.
pink, right? So even they recognizes all this thing. So depending upon the
Okay.
color, okay, if we are having a rose plant of red color, so the plant is going to accept the pollen grains of red color only, right?
Okay.
And the right type is what the style is going to identify. Oh, this is a red color pollen grain. I'm also growing a red color pollen grain. So let's absorb it now. Let's start providing nourishment to this pollen grain. And after giving nourishment to that
Okay.
pollen grain, what is going to happen? That pollen grain is going to absorb the nourishment, that is a sugary substances. And sugary
Yes.
substances is nothing but it is a starch, and that pollen grain is going to swell out. And after swelling out, what is going to happen? The pollen tube, the germ tube has been supposed to get germinated, right?
Yes.
And then the further double fertilization activity is going to start. But what if that pollen grain is of wrong type? The pistil means what? The pistil, it is nothing but it is our own stigma. So stigma is also
Yes.
called as an pistil.
Pistol.
Understood? Yes. So therefore, this pistil means the stigma is going to reject that pollen grain. So what it is going to do? If it is rejected, so germination activity will carried out? No,
No.
because the fertilization will not occur. Hence, the nourishment will not be provided to that pollen grain, because it is not ours pollen grain, it is some others pollen grain which has been deposited on our plant.
Yes.
Understood? So no germination. Firstly, no nourishment. So no germination. Nothing is going to happen. That what
Yes.
happened? That pollen grain is going to die on that stigma and it is going to get degenerated. Okay. But
Yes.
whenever the right type is happening, compatibility of pollen grain on stigma, okay, and pollen tube comes out through one germ pore. Means whenever if the plant is having a right pollen grain, we everybody know what is going to happen. The germination process is going to happen. Why? Because compatibility means what? It is our pollen grain. So we all are familiar, we
Yes.
both are familiar with each other. So let's
Yes.
do the fertilization activity and germination activity, and the germ pore is going to start coming out. That is, the germ tube process is going to start. Okay. Now what
Yes.
will happen? The content of the pollen grain, whichever it is consisting of, it is start going to move via the pollen tube. And this pollen tube, which is present from stigma through the style to the ovary, means what is
Yes.
going to happen? That pollen tube, which is the pollen grain, which is present, it has absorbed the nourishment, it has swelled. So what it is going to do? Let's send whole of our part, let's send whole of our content from the pollen grain via the pollen tube. And from that pollen
Yes.
tube, it is going to pass from the style, and from the style the main aim is to reach towards our ovary.
Ovule.
Yes,
Ovule.
yes, yes, towards the ovule, right?
Ovule.
Yes. So that ovule and then towards the embryo sac. In this way, this interaction has been carried out. All right. Now, later
Yes.
on, wherever the plants, the angiospermic plants which are present during germination, this pollen grains divides. We everybody know, right? Even we have studied in
Yes.
microspore and male gametophyte. This pollen grain
Yes.
divides and it forms its two cells.
Two cells.
Yes, the cells are the generative cells and a vegetative cell.
Vegetative cell.
Yes. Now this generative cell will get divided and it will form its own two male gametes.
Yes.
Now you understood from where the male gametes came, Akansha?
Yes.
Okay, so this male gametes have been from our generative cells, which we have already saw in male gametes, right? After
Yes.
that, what will happen? After reaching towards the ovary, the pollen tube is going to get ruptured. Okay, it is going to get disturbed, and the wall of the ovary it is going to touch to the ovule. The normal process which you
Okay.
and I know, right? And this pollen
Yes.
tube is going to enter to the ovule because we are studying an anatropous ovule. So the what pollen tube is going to enter via micropylar end, micropyle region,
Yes.
which is present down towards because we are studying the anatropous ovule,
Anatropous ovule.
right? And hence the
Yes.
growth will get continued. And what will happen? The embryo sac, via it will go to the embryo sac. First, it is going to rupture the nucellus, and from there, it is going to enter to the embryo sac, from through which the one synergid is going to form a filiform. Okay, filiform
Yes.
apparatus, and it is going to release a male gametes.
Male gametes.
Yes, what it is going to do, it is a normal process, right? Which we have already saw in female
Yes.
gametophyte. What it is going to do, it is going to come into embryo sac. Once the embryo sac is ruptured, it is going to meet to the egg cell, right? And the
Yes.
synergids are going to provide a nourishment for the development, isn't it? And after
Yes.
doing that, the filiform means what? The synergids itself, it is consisting of an egg apparatus, right? What is an egg
Yes.
apparatus, Akansha? Egg cell and synergids.
Yes.
Okay. So whenever the egg cell and synergids are playing role, we everybody know it is called as an egg apparatus.
Egg
But
apparatus.
yes, but whenever we are having a filiform apparatus, means the synergids have been played role. And after
Okay.
that, it is going to happen what? Already the male gametes are present, and those male gametes are going to release into our the
Release.
embryo sac. बराबर.
Yes.
तो वो मेल गैमेट्स एम्ब्रियो सैक में रिलीज होने के बाद आपको भी पता है एक मेल गैमेट जाके फ्यूज होगा, किसके साथ जाके फ्यूज होगा?
Excel.
एग सेल के साथ, एंड इट इज गोइंग टू फॉर्म व्हाट?
Diploid zygote.
Very good. And the second one, it is going to fuse it
Secondary nucleus.
and it is going to form.
Triploid primary endosperm nucleus.
Very good. So this process is going to happen into the pollen pistil interaction. The basic motto of the pollen pistil interaction is how to identify our own pollen grain.
Okay.
Okay, and after identifying the own pollen grain, what are they going to do? What are they going to, how they are going to release, how the male gamete is formed, and after forming, how this double fertilization activity has been carried out. Understood, Akansha?
Yes.
If this thing is cleared, then the whole chapter is cleared of yours.
Yes.
All right. So you can understand the normal mechanism which is happening into the angiospermic plants. This happens in
Yes.
each and every angiospermic plants. Every day,
Okay.
every minute, and every second.
Okay.
But only during the day process. All right.
Yes.
Yeah, because we everybody know that during the night process, the stomata is closed.
Closed.
Transpiration activity is low, right?
Okay.
Yeah. So in each and every process, this is carried out during whom? During the day process.
Day.
Till the sun is there, till that time, this all this process is going to be carried out. All right.
Okay.
Now let us move towards our next term, that is development of the endosperm. See, Akansha, I have kept all this concept in a clumped format. Why? The
Yes.
reason behind that is they all are interlinked with each other, right? If in
Okay.
between, if I'll start with a pollination, and I'll leave this all such kind of in part, and whenever I'll teach you with the endosperm, or I'll go with a double fertilization, or I'll go with for like female gametophyte, I have to revise it back, right? These all are
Okay.
interconnected with each other. So when the things are interconnected with each other, it enjoys you also and me also to understand the lecture, and the lecture to enjoy also, right? So that's why
Yes.
I have kept everything in a clumped manner, so that it is easier for you all students to understand, right? Now let us
Yes.
see the development of endosperm. Now we everybody saw that what into endosperm. All right. So this endosperm which we have saw, where am I? Yeah. So this endosperm, it is what basically it is providing a nourishment, right? What it is doing, it is
Yes.
providing a nourishment to the,
Nourishment.
yes, it is giving a nourishment to the seed, isn't it? Now we saw
Yes.
here, isn't it, triple fusion activity, right? The second male gamete which has been fused with a female gamete, isn't it? What it has been
Yes.
done?
Endosperm of seeds.
Yes, it has formed a endosperm of seed. And this endosperm of seed, it provides nourishment to the embryo sac, that is to the embryo of the seed. Right, Akanksha?
Yes.
All right. Now, why I came on this slide? So that you should know what is happening and what not. All right, from where this embryo came. Now,
Yes.
yes, the endosperm has been developed from TPEN. We both know that. Now, this
Yes.
primary endosperm, okay, this primary endosperm nucleus, it every time divides and redivides. Why does it divides and redivides? Basically, to form a tissue which is called as an endosperm,
Endosperm.
right? Now, this endosperm growth rate is very faster than embryo, okay, and it grows within a shorter period of time, all right. And this embryo, what does it do? It surrounds the, sorry, this endosperm, it surrounds the embryo, okay. Again I
Okay.
am repeating, comparison to embryo, the endosperm growth rate is fast, all right,
Yes.
and this endosperm is surrounded by the embryo, all right.
Okay.
And the main function of the endosperm is to provide the nutrition, that is nourishment, provide food material to the embryo,
Okay.
that is to the embryo sac.
Yes.
Yeah, Kansha. Now, what
Yes.
happen in some plants? This endosperm is completely utilized. Okay. What happened in certain plants, this endosperm which is present, what does the plants do? Plants use fully those endosperm. And after using it fully, it gets starting developed into the embryo. Okay. And later on, when you will see, this embryo is not present. Why it is not present in some of the plants? Because this endosperm is not present. Why it is not present? Because the embryo which is present, the embryo sac which has been present, it has been fully utilized by the endosperm, all right. For example,
Okay.
like one day what happened, we everybody, what are we doing? We are just using the stationary material. Now you and I, we are doing a project, and we are using, constantly
Yes.
using all the study material, using. We are not going into a stationery to buy a new stationery, right? We both are using.
Yes.
And after finishing of our project work, or after little bit our project work is remaining, we both realize, oh my god, the stationery has got over.
Yes.
Right. So what happened? This process is happening into these plants also. In some of the plants, they're just using the endosperm. Chalo, we will use it. And the embryo is
Okay.
formed. And after the embryo is formed, when the embryo is requiring a endosperm for a nourishment, so what happened? They suddenly realized, oh, there is no endosperm left. Because we have what? We have utilized
Utilized it.
the whole embryo. Okay. And what
Yes.
happened? Later on, they realized that our whole process have been completed. There is no endosperm present. The embryo has got mature. The seed has got mature. Why it is like that? Once the embryo has been completed, it functioning, it developing process, definitely the seed has got mature. Because inside the seed, there is a presence of an embryo. Okay, and
Yes.
such seeds which are utilizing whole of the endosperm, the seeds are without endosperm, and they are called as in non endospermic seeds.
Okay.
And what are the example? The example is sunflower, pea, that is
Pea
green pea,
groundnut.
and groundnut. Understood? Why
Yes.
this is non-endospermic? Because the endosperm has been fully utilized by the
Utilized by embryo.
embryo. Okay, and one day they suddenly
Yes.
realize, oh my God, our seed has got mature, our embryo has got mature, but there is no presence of an endosperm because
Endosperm.
we have utilized it.
Utilized it.
Yes.
Yes.
Okay. In some plants, if you will see, the endosperm is partially utilized. Means what? It is little bit utilized during the
Okay.
development of the embryo. And what happened? What does it results in? It results in endosperm as a mature seed because some of the embryo is sorry, endosperm has been utilized. Okay, certain part of
Yes.
the endosperm is still remaining. Okay, and still the certain part of the endosperm is remaining, but the seed has got mature. So whichever that endosperm part is remaining, what that seed uses its head. Seed says, okay, अभी ये बचा हुआ एंडोस्पर्म है, मैं इसको ये मुझे नरिशमेंट दे रहा है, तो मैं एज अ फूड मटेरियल के तौर पे इसे मैं कहीं पे स्टोर करके रख देती हूँ. Right?
Okay.
So that
Endosperm.
Yes, endosperm. Because
Okay.
see, what we said in some plants, endosperm is partially utilized. Partially utilized. Barabar?
Yes.
Partially means what? Little bit it has been used and
Utilized.
yes, little bit it's still remaining.
Okay.
Okay. Half of the part is used, half of the part is still remaining.
Okay.
Okay, means what? That seed has played a brave role. Okay, now what is the thing? Whenever we have a more savings of hours, if we are not working, or we are just sitting at home, what it is going to do? It is going to help us. That saving is going to help us, isn't it? That what?
Yes.
The saving is going to help us. Though we are not working, though we are not doing anything, but our saving is strong, so we can sit at home and we can eat it, isn't it? How much ever is required, we utilize. How much ever is there, we are keeping it, isn't it? The seed is also using its own mind. They are not like a non-endospemic seeds. Okay, कि सब कुछ यूटिलाइज कर लिया, सब कुछ खत्म हो गया, अब हम मैच्योर हो गए. Right? All right. It is same like an what? It is same like an a retirement, isn't it? The
Okay.
seed has retired, the seed has got matured, but still that seed is having the saving, that is
Savings.
a endosperm. It is having a
Yes.
little bit saving, little bit endosperm. So that seed has used its mind. That is what I'm going to keep this little bit of my saving, little bit of my endosperm.
As food material.
Yes, as a food material. Understood? So
Yes.
that endosperm is what? That endosperm is quite talented. All right, and it is quite good.
Yes.
Okay. Now, Akansha, this is a type of an endosperm, right? This is nuclear, cellular, and halobial. We are soon going to study the types also. All right.
Okay.
Only the thing is, you just look and you keep it, and soon we will be studying this type in detail. All right.
Okay.
Yeah. Now, so such seed with endosperm. Okay, now these seeds are consisting of an endosperm because though
Yes.
the endosperm is little, but still this
Yes.
endosperm they are having it. They are called as an endosperm.
Endospermic seeds.
Yes, example is custard and maize. Okay, now
Yes.
depending upon the mode of development, depending upon how this endosperm get developed, hence it has been divided into the different types. The first type of the endosperm is nuclear endosperm, right? We called as a nuclear endosperm. Second type is cellular endosperm, and third type is what? Third type is
Fallopian.
fallopian endosperm. Okay, so now let's begin. So the
Yes.
same diagram I said you. All right, this three diagram which you are observing it, this is of nuclear. This
Okay.
three diagram, it is of cellular, and this two,
Cellular.
Hello.
Okay.
Question expected into the exam. In exam, the question can be expected for a long answers, that is development of endosperm. If they are asking development of the endosperm, Akansha, you have to write down the types also.
Okay.
Second question which can be asked into a board exam, that is what they can ask you, that explain only the types of endosperm. So we have to write down about nuclear, halobial and cellular endosperm. Okay.
Okay.
And sometime it is asked into the answer in one sentence, or definition, that define nuclear endosperm, or what is halobial, what is
Okay.
non-endospermic endosperm or endospermic endosperm, right?
Okay.
Sometimes the question is as asked, that is what, give the examples of an endospermic seeds. So you can write it down. The endospermic seeds example is castor and maize, and
Yes.
non-endospermic is,
Sunflower, peas,
yes, sunflower,
and
pea,
groundnut.
and groundnut. Very good. Now let us begin with our further things, that is the types of endosperm. The first type of the first mode of development, depending upon the mode of development of endosperm, the first type is nuclear endosperm. This nuclear endosperm, it is one of the most common type of an endosperm, right? In this, the primary endosperm nucleus means the first nucleus which is formed into the endosperm, it undergo the nuclear division. अगेन आई एम
Okay.
रिपीटिंग इट आकांक्षा, जो पहला न्यूक्लियस हमारे इसमें फॉर्म होता है, एंडोस्पर्म में फॉर्म होता है, उसे हम कहते हैं प्राइमरी. प्राइमरी,
Okay.
सेकेंडरी, टर्शरी. प्राइमरी मीन्स फर्स्ट, secondary means second, tertiary means
Second.
third,
Third.
right? So the first nucleus which is formed into the endosperm, and
Yes.
it is going what it doesn't require any kind of a cell for conducting this, what for conducting this nuclear division. It can go freely under any kind of a division, right? It is what it the cell is
Yes.
free, the nucleus is free to go under what, to go under the nuclear division. So hence it is called as in what? It is called as in karyokinesis. Whenever the cell undergo the nuclear division, right? So it is called as in karyokinesis, right? The cell has been divided. The cell division takes place into how many processes, dear?
Two.
Yeah, name them.
Mitosis and meiosis.
And that mitosis and meiosis are depending upon whom?
Depending upon
Yes, means before the mitosis and meiosis and mitosis, what are the first processes? Depending upon them, the mitosis and meiosis have been started. That is karyokinesis and cytokinesis. Okay. Now you should remember this, Akansha, that this cell division, definitely we have a mitosis and meiosis. Okay. But this mitosis and meiosis also, from where it came from? It came from the karyokinesis and cytokinesis. Okay. So whenever this karyokinesis, whenever the nucleus is undergoing the nuclear division, so it is called as in karyokinesis. जहाँ कहीं पे भी न्यूक्लियर डिवीजन हुआ तो आपको पता रहना चाहिए वो कैरियोकाइनेसिस. कैरियो इज इक्वल टू न्यूक्लियस, काइनेसिस इज इक्वल टू डिवीजन.
okay.
Are you getting my point, Akansha?
Yes.
Yes. Cytokinesis. In this nuclear division, the cytokinesis is not present. So the nuclear division is not followed by a cytokinesis.
Cytokinesis.
Yes, cyto is equal to cell, kinesis is equal to division. Understood. So that's why
Yes.
I ask you, if you will open the 11th standard test book, Maharashtra State Board, when you will refer the chapter of in cell division, dear, you will see
Yes.
after the interphase there is a presence of an how many stages? What are the types of in stages of cell division? So they are the
Yes.
karyokinesis and cytokinesis. Yes.
Cytokinesis.
And depending upon the karyokinesis and cytokinesis, the M cycle is depend. What is that M cycle? M cycle means our mitosis and meiosis. Okay. So these are certain things, right? So now we are talking about nuclear endosperm. So this nuclear endosperm which is present, so always remember the karyokinesis is supposed to be happen, right? Why? The region behind that is what? The region behind that is the nuclear endosperm. We are studying about nuclear. So the
Yes.
karyokinesis is going to happen. There will be no cytokinesis activity will be conducted. Understood,
Okay.
Akansha? Now, that's why it has been said nuclear division is not followed by cytokinesis. In this results, what? In this, the endosperm, in this, the nuclear endosperm, the large number of the triploid nuclei are freely formed. Therefore, later on they are suspended and it has been what? It has been commonly seen into the cytoplasm. Right? Now, one thing we
Okay.
should remember, whenever this nuclear endosperm is carried out, there will be a large number of the what? How many? Triploid means the three sets of the nuclei is going
Three.
to form. Right? And what are they are
Okay.
going to happen? Later on, they are going to get suspended. Suspended means wherever they are present they are going to be constant on their places, okay, and they will be
Okay.
commonly found into the cytoplasm. Cyto is equal to, Akansha, just now I said,
Cell.
cell. Plasm means it is a house where all type of the cells are formed,
Yes.
right? So what? It
Yes.
will be a part of the cytoplasm. This triploid nucleus, these three set of a nucleus, they will be a form, they will be a part of that cytoplasm. All right. And later on,
Okay.
the wall formation is occurred. Means the endosperm wall formation has been taken place very well properly. All right. Now you understood
Okay.
why there is a presence of in three cells, three nucleuses over here.
Yes.
Okay. This is what? This is a young nuclei, right? This
Yes.
is this nuclei have been went little bit under a division. That's why you can see there is a presence of an embryo in this. Okay. Over here, if you will see only the
Okay.
oospore is present. Oospore is
Yes.
which? Oospore is a young cell of this nucleus, right? And
Okay.
this later on, this oospore, it has been went under the further cell division and it has formed what? It has formed a embryo. And this embryo, if you
Okay.
will see this whole cell where my cursor is rotating there, if you will see over there, you will see this mature nucleus have been formed. Mature nuclear endosperm has been formed with the presence of an wall also, right? See the
Okay.
outer wall. Can you see this outer wall where my
Yes.
cursor is rotating? Yes. And
Yes.
inside that we have a many more cells, different cells. So this is what? This
Okay.
is a cytoplasm, isn't it?
Okay.
Now, in
Yes.
this, if you will observe, these are the big cells which we are seeing, right? We are looking towards the larger
Yes.
cells. But same thing when I will show you under a compound microscope, this will be what? This will be a minute cells which you will found which are under a triploid condition, right? And I'm
Okay.
talking about cells, sorry, it is a nucleus. These are the three triploid nucleus.
Okay.
Okay, all right, Akansha.
Yes.
Understood what I am speaking about. The
Yes.
first endosperm type we have studied, that is nuclear endosperm. In this, the nuclear division has been carried out, the karyokinesis process has been carried out.
Yes.
Cytokinesis is not followed.
Not followed by it.
Yes, and after that, even we have saw, therefore, there is a formation of an triploid nuclei. The
Yes.
reason behind that is, sorry, that is what? The primary endosperm, the first nucleus which is formed into the endosperm, it has undergone the free nuclear division. None of the cell or none of the nucleus is required to the single endosperm nucleus. It has went alone under the nuclear division. That's why the karyokinesis has been followed,
Okay.
and this karyokinesis have formed a large number of triploid nuclei. And later on they have been constant. That's why I have written they have been suspended into the common cytoplasm. means into a whole
Okay.
endosperm, which is nuclear endosperm. In this, you will found that three of this nuclei, which are present, they extend. And later on, the wall formation is also done. The wall formation is also occurred, right, into
Yes.
nuclear endosperm. Understood, Akansha, the first type there?
Yes.
Now let
Yes.
us move towards a second. Okay, second is a cellular endosperm. It is a very less and mostly seen into a dicot family plants. What is a dicot?
Okay. Two
The
cotyledons.
plants which are having a two cotyledon means two seeded
Yes.
plants, right? The
Yes.
plant which is consisting of in two seeds, isn't it? So in
Yes.
this it is very less common, means very rarely, not rarely, very I can say little bit it is seen into a plants, but mostly this cellular endosperm you will see into a dicot. Okay, dicot family. Now, in
Okay.
what does happen in this primary endosperm nucleus, it undergo free nuclear division. Okay, so
Okay.
definitely there is a karyokinesis activity has been carried out. But what
Yes.
nuclear division said, I'll not go alone for division. I'll take all these cells for with me for the division. That's why what is that
Okay.
being said? Nuclear division is also followed by cytokinesis.
Cytokinesis.
बराबर, क्योंकि यहाँ पर सेल्यूलर
हाँ.
एंडोस्पर्म होना, क्योंकि हमें ऐसा सेल चाहिए जो कि कैसा है, जो कि सेल से भरा हुआ है, ना कि न्यूक्लियस से. न्यूक्लियस तो है लेकिन सिंगल न्यूक्लियस प्रेजेंट है, लेकिन बाकी सारे सेल्स है उसमें.
Okay.
आया, समझा आकांक्षा?
Yes.
ओके.
हाँ.
इफ यू विल ऑब्जर्व इन दिस न्यूक्लियर, बराबर, अगर आप न्यूक्लियर में देखते हो तो पूरा जो हमारा एंडोस्पर्म है वो न्यूक्लियस से भरा हुआ है, बराबर. उसमें तीन
हाँ.
न्यूक्लियाई फॉर्म हुए हैं, मतलब हुआ एक, लेकिन वो जो तीन जो है वो ट्रिप्लॉइड कंडीशन में है, ट्रिपल सेट है
हाँ.
उनके पास, बराबर. लेकिन यहाँ पर क्या है सेल्यूलर में, इन सेल्यूलर इट इज व्हाट, one nucleus is present because karyokinesis activity has been done. But what nucleus said, I'll not go alone, you everybody should come with me, each and every cell should participate. So that's why the karyokinesis is followed by a cytokinesis.
Cytokinesis.
Understood, dear? And the
Yes.
example is what? The example is balsam and adoxa.
Adoxa.
Okay, these two are the
Yes.
plants. Okay, these are not Indian species, they are the
Okay.
uh, foreign species. So therefore mostly the cellular endosperm has been seen into the balsam and adoxa. All right, these two are the
Okay.
plants which are common. All right. Now, the thing is that now you will be thinking that, ma'am, over here again there is a tree. Yes, there is again the
Yes.
same process it has been carried out, how it has been carried out into a nuclear. Barabar. Therefore,
Yes.
I said you that the cell is whole consisting of a nucleus, but into a cellular, triploid cells are formed. Okay, if anybody asks you, if in exam it has been asked that how many cells are formed into a cellular, you have to say triploid cells. Nuclear
Okay.
endosperm, triploid
Triploid.
nuclei. Triploid nuclei.
Nuclei.
Barabar.
हाँ.
And in cell, you have to just write, instead of nuclei, you have to write it down triploid
Triploid
cells.
cells.
Understood? So therefore,
Yes.
that's why over here the three cells are formed. And same way you can see there is a formation of an oospore, okay, young cell. After that, there is a formation of an embryo and endosperm, right? Then the
Yes.
mature cell is formed with a cell wall. Understood?
Yes.
Okay, dear. All right. Now let's move towards further, that is halobial endosperm. The third type of an endosperm is halobial endosperm. It is very commonly found into a monocot plants. Mostly found in which of the plants? The Halobiae series. Okay. Now,
Okay.
into our test book, if you will refer in 11th standard test book, classification when we see that is what, kingdom, division, subdivision, class,
Yes.
subclass, family,
Yes.
subfamily, series, order, genus, species, right? This is a sequence
Yes.
of the classification, isn't it?
Yes.
When we see the first chapter, that is a diversity of organisms in FYJC, that is in 11th standard. Okay,
Yeah.
so now we know. Now, family, if we talk about this, Hellobial is what? Hellobiaceae, right? But if you will see the
Okay.
series, that is Hello Bee. Barabar? This Hello Bee series, mostly this plants Hello Bee, they are coming under a monocot plant. So you can take any monocot plants. Monocot plants means which plants,
Okay.
Akansha? Belonging to which family?
Rice.
Yes, the leaves
Rice.
are looking
Paddy.
like, yes, paddy fields. So ra- leaves are like what kind of how the leaves look like?
Look like as in.
You remember I said you Grassaceae family?
Yes. Yes.
Yes, the leaves seems
Which
to be
looks
like
like
grass.
grasses.
Yes, that is a, certain things we have certain, like a nickname we have. In the same way, we have a nick conditions. Okay, we have a
Okay.
nicknames for a plants or for a certain conditions or the certain words to remember, right? We cannot remember togetherly.
Okay.
Monocot, dicot, right? So normally students get confused. Oh my God, this is
Yes.
monocot, this is dicot. From where we should remember? So the easier way for the monocot is
Yeah.
the plants which are having a resemblance like a grass, right? Mostly they are belonging to the Grassaceae family. So you can
Yes.
say they are the monocots. So this Hellobial, you will see all under the
Okay.
grasses. The leaves we are having the leaves which are having a grass-like resemblance. Okay, that is a monocot family. Hmm.
Okay.
So the first division of primary endosperm, that is the first primary nucleus, the division it is going to happen. The first division, it is followed by transverse wall, and the cell which it has been divided, it will be dividing unequally. Okay, again I am repeating it. First division which is carried out into this family, that is a Hellobial endosperm. We everybody know whether it is nuclear, whether it is cellular, right? Whether it is Hellobial, we have a first nucleus which is formed, which we are calling it as
Yes.
an primary endosperm nucleus. What is going to happen?
Yes.
There is what it is going to happen. There will be a division activity which will be carried out, and the division activity, it will be done first, it will be done for a wall. Okay, now what is a wall? Now,
Okay.
yeah, you can see, Akansha, this is a mature nuclear. This is a
Yes.
nuclear endosperm. This is a mature. Outer wall is present here. Outer covering is present
Yes.
here. Barabar. This oval shape, this round shape, we called it as an outer wall. Inside that,
Yes.
all the nucleuses are present. Now, this one,
Yes.
which is an irregular shape of cellular, this is what? This is the outer wall, which is an outer covering, isn't it?
Yes.
Inside this, all the cells are present, including embryo and endosperm, isn't it? In the same
Yes.
way, in this Hellobial, we have a outer covering, right, dear?
Yes.
Barabar. Now, how many this is? Two. One,
Two.
over one, two. Barabar. One, and this is two.
Two.
Right? Huh. Now, what my
Yes.
condition is, what my sentences says, that the, see, first division is happening. Nucleus, the
Yes.
first nucleus which is formed, it has been went under the division, right? Followed by wall division. Wall division means what, Akansha? You can see, this is what? This
Yeah.
is a
It is
covering.
dividing.
Yes, this coverings, this outer covering means what? This outer wall, they have been under the division. Understood? That's why
Okay.
these two divisions, these two walls have been formed. So इसके लिए न्यूक्लियस तो गया डिवीजन में, लेकिन
हाँ.
साथ में इसके वॉल्स भी डिवीजन में गए.
Okay.
Okay, now when this division have been went, this wall has been went under division, therefore this cell divide unequally. Do this cell have a shape? Can you see both the cells?
Yes.
Are they are in a proper shape, Akansha?
No.
No. बराबर. मीन्स सेल
हाँ.
क्या हुए? अनइक्वली डिस्ट्रीब्यूट हुए. समझ में आया? इनको प्रॉपर
हाँ
शेप नहीं है। अगर मैं आपको कहूँ न्यूक्लियर का शेप बताओ बेटा, तो आप कहोगे मैम ये ओवल शेप में
हाँ ओवल
है कि नहीं। आप सेल्यूलर का शेप मुझे बता सकते हो?
ओवल
सेल्यूलर एंडोस्पर्म का आप मुझे बता सकते हो?
नहीं
नहीं, बराबर है कि नहीं। आप मुझे
हाँ
हेलोबियल का बता सकते हो?
नहीं
नहीं. So what has happened? The wall have been divided, but they have divided according to that way. They have been unequally divided. मतलब unequally मतलब देखो शेप नहीं है, कहीं पे ज्यादा डिवाइड है, कहीं किसी को ज्यादा पार्ट मिला है, किसी को कम पार्ट मिला है, बराबर।
हाँ
यहाँ पर भी बाजू वाले में भी आप देखोगे जहाँ पे एम्ब्रियो फॉर्मेशन हुआ है, किसी किसी को ज्यादा पार्ट मिला, किसी किसी को कम पार्ट मिला, right? So the cells which
हाँ
has been divided, what are they saying? They have been divided unequally. Understood, Akansha?
ओके
Okay, there is no proper
यस
proportion of the part has been distributed.
यस
Okay, half of the part has been in certain part it has been distributed more, in certain part it has been distributed less.
लेस
Okay, now the smaller cell is called as an chalazal cell, and the larger cell is called as a micropylar cell. Understood?
ओके
Means the smaller part which is present into this halobial, it is called as an chalazal cell, and the larger part which is present, it is called as an micropyle.
माइक्रोपाइलर सेल
ओके, माइक्रोपाइलर सेल। समझ में आया?
यस
बराबर। ये दो जो
हाँ
अनइक्वल में डिस्ट्रीब्यूट हुए, छोटा कौन और बड़ा कौन, हु इज स्मॉल एंड हु इज लार्ज।
चलाजल माइक्रोपाइलर
Yes, yes. Okay. Now,
यस
later on, free nuclear division, the wall developed between nuclei in micropylar chamber. Okay, what they are saying, the half of the part where they have divided the both cells which are divided via nuclear division, the wall has been divided according to that way between the nuclei. And what they have done, they have formed a chamber between a micropylar. Okay, what the
ओके
things was supposed to happen, that the सबसे ज्यादा मेजर पार्ट जो आप देख रहे हो वो क्या है, माइक्रोपाइलर के अंदर है, बराबर। तो उन्होंने
हाँ
क्या कहा कि जो कोई भी हमारा डिवीजन हुआ, वॉल डिवीजन हुआ, डेवलप्ड हुआ, वो न्यूक्लियाई के बीच में हुआ, राइट। लेकिन
हाँ
ज्यादा से ज्यादा पार्ट किसके लिए बना, माइक्रोपाइलर एंड के लिए बना, मतलब माइक्रोपाइलर चेम्बर के लिए बना। समझ में आया आकांक्षा?
हाँ
एनीथिंग विद एनी काइंड ऑफ
हाँ
डाउट
हाँ
टिल योर डियर? एनी डाउट डियर टिल योर?
यस यस
Okay, do you have any kind of in doubt? Can you hear me?
हेलो
Hello Akansha, can you hear me?
यस यस
Yeah, is till now is everything is clear?
यस
Okay, shall I move further?
यस
Okay, it is an intermediate between cellular and nuclear endosperm. What is the meaning of an intermediate?
इन बिटवीन
Yes. So the partition has been done between whom? Between the cellular and the nuclear
न्यूक्लियर
endosperm.
एंडोस्पर्म
But अगर आप ये देख रहे हो तो ये दो पार्टीशन हुए हैं, right? So this two
हाँ
partition is between. So this two intermediate is between whom? Between the nuclear and whom? Between the cellular endosperm.
सेल्यूलर एंडोस्पर्म
Understood, Akansha?
यस
तो अब इन दो में डिवाइड हुआ, that's why we called it as an halobial endosperm.
ओके
Okay, when the nucleus, when the
यस
nuclei was divided, we called it as a nuclear endosperm. When the cell was divided, we
यस
called it as a cellular endosperm.
सेल्यूलर
But yes,
एंडोस्पर्म
but when the division was done between both the cellular and nuclear, we called it as an halobial endosperm.
हेलोबियल एंडोस्पर्म
समझ में आया? Okay. All
हाँ
right. So the example
यस
again, it is not indigenous species, that is, it is not Indian species, it is a foreign species. Hence the name of the Plant is asphodelus.
Asphodelus. Okay.
Okay, so this all are the three
Yes.
types of and mode of, depending upon the mode of development of endosperm, these three types of and what these three types of an endosperm are dependent. That is, the first one is your
Okay.
nuclear endosperm, second one cellular, and third one is
Cellular.
halobial endosperm.
Hello, Bill.
Understood,
Yes.
Akansha? This development of
Yes.
endosperm wholly you understood?
Yes.
Any kind of in doubt till here?
No.
Shall I move further?
Yes.
Okay. Now, pollination and types and its agencies. Okay. Now, into our
Okay.
nature, there are different types of an pollination which has been carried out. What is the meaning
Yes.
of an pollination, dear? Transferring of or carrying of pollen
Pollen
grains.
grains.
Yes, from one plant to the another plant, right? It is called as an
Yes.
pollination. All right.
Pollination.
Yes. So the common leaf, since from lower grades we have saw the pollination has been carried out by a wind, that is our air, or it has been carried out with the insects. Am I
Yes.
right? Now, this pollination, if the proper definition is asked, what is a pollination? So pollination means what? Transfer of pollen grains, okay, from anther to stigma of same flower is called as an pollination.
Pollination.
Okay, again I'm repeating. When the pollen
Yes.
grain is transferred from anther to stigma, okay, of the same flower, means of the same plant, it is called as an pollination.
Pollination.
The pollination is of two type. The first pollination is called as in self pollination, and second one is called as in cross pollination.
Cross pollination.
Okay, now this self pollination means what? When these pollen grains are transferred from anther to stigma, okay, the stigma of the same flower or another flower present on the plants of same body. Again, I'm repeating it. Self pollination is what? Self pollination means when the pollen grain, जब कभी भी pollen grain जो कि एंथर पे प्रेजेंट है, बराबर, स्टिग्मा पे, ओके, एंथर टू दी स्टिग्मा, एंथर से स्टिग्मा के पास जाएगा, लेकिन आइदर वो सेम फ्लावर का होगा, राइट, या फिर अनदर फ्लावर का होगा, दूसरे फ्लावर का होगा, लेकिन सेम of the plant body.
Okay.
फॉर एग्जाम्पल, रोज प्लांट है, बराबर,
हाँ.
इस रोज प्लांट पे अब सेल्फ पॉलिनेशन मतलब खुद पे ही पॉलिनेशन होना, बराबर। अब ये रोज प्लांट पे क्या
हाँ.
हुआ, उनके पोलन ग्रेन्स क्या हुए, अभी फ्लावर है, डेफिनेटली एक इस पे एक ही फ्लावर नहीं आना है, बराबर, दो चार फ्लावर्स आते हैं, ज्यादा फ्लावर्स आते हैं ब्रांचेस पे,
हाँ.
बराबर है। सो उस फ्लावर पे क्या होगा हमारा, वहां से जो पोलन ग्रेन्स डिपॉजिट हुए, तो वो पोलन ग्रेन्स का कहाँ से ट्रैवल है, एंथर से लेकर स्टिग्मा तक ही वो जाएंगे, बराबर।
हाँ.
लेकिन एंथर से लेकर स्टिग्मा तक ट्रैवलिंग होता है पोलन ग्रेन्स का, राइट। आइदर वो
हाँ.
सेम फ्लावर पे होंगे, बराबर, मतलब वो जो फूल है वो क्या है, वो सेम झाड़ पे ही प्रेजेंट है, सेम प्लांट पे ही प्रेजेंट है, ओके।
हाँ.
सो वो, नाउ दिस इज अ प्लांट, बराबर, ये मेरी यहाँ पे रोजेस है, ऑल द रोजेस आर प्रेजेंट, राइट। सो
Yes.
हाउ एंथर टू स्टिग्मा इज अ ट्रैवलिंग पाथवे ऑफ पोलन ग्रेन, राइट। सो ये पोलन ग्रेन उड़ा बाजू के फ्लावर पे, अंडरस्टूड,
Okay.
मीन्स व्हाट
हाँ.
हैपन, सेम प्लांट है लेकिन बाजू बाजू में ही उड़ रहा है,
Okay.
ओके, मीन्स खुद पे ही
हाँ.
पॉलिनेशन हो रहा है,
हाँ.
understood, Akansha? So this is called as in
Yes.
what? This is called as in self pollination. What it is called as in self pollination.
Self
That plant,
pollination.
yes, that pollen grain haven't traveled some other rose plant for the pollination purpose, understood, within same
हाँ.
plant पे ही हुआ,
Okay.
so it is called as in self pollination, same body plant पे ही हुआ, समझ में आया?
हाँ.
Hmm. if it would be happened on some other plant, okay, same species के ही दूसरे वाले रोज प्लांट पे उड़ के गया, तो हम उसे सेल्फ पॉलिनेशन नहीं कहते,
Cross pollination.
क्रॉस पॉलिनेशन कहते. समझ में
Okay.
आया? अंडरस्टूड
हाँ.
टिल हियर? ओके.
Yes.
नाउ this self pollination is of two types. First one is autogamy, second one is geitonogamy.
Okay.
First one is autogamy, second one is geitonogamy. Autogamy means what? Transfer of pollen grain from anther to same flower. एक फ्लावर है, एक फ्लावर पे ही काफी सारे एंथर्स है, काफी सारे स्टिग्मा है, वहीं पे पॉलिनेशन होगा.
हाँ.
ओके, इट इज कॉल्ड एज एन ऑटोगैमी. एग्जाम्पल इज ग्रीन पी प्लांट.
हाँ.
ओके, सेम फ्लावर है, सेम फ्लावर वो लोग कष्ट ही नहीं ले रहे हैं कि हम दूसरे कोई प्लांट पे या फिर नीचे वाले प्लांट पे जाके हम अपना पोलन ग्रेन्स गिराए.
Okay.
Okay, they are not taking that kind of an, they are not doing that kind of an hard work. They are just transferring to just nearby people, or on a within
Yeah.
the same flower they are
Same.
transferring. And this is just
Yes.
called as an autogamy. What is the meaning of an geitonogamy? The transfer of pollen grains from anther to stigma of another flower present on same plant body. मतलब सेम
हाँ.
प्लांट है. ओके, ये क्या कर रहे हैं, बाजू के भी जो फ्लावर्स है सेम प्लांट्स पे ही, आजू बाजू वाले जो फ्लावर्स है उसी पे, एक ही फ्लावर तो नहीं आने वाला है एक रोज प्लांट पे,
हाँ.
काफी सारे रोजेस आएंगे,
हाँ.
right? So what are they doing? They are present on a same plant body and they are distributing to the other rows also. समझ में आया?
Okay.
सेम
हाँ.
प्लांट है, लेकिन जितने कितने भी रोज हुए उन्हीं के बीच में पॉलिनेशन एक्टिविटी हो रहा है. It is
Okay.
called as an geitonogamy.
Geitonogamy.
Yes. What is the pathway? The pathway is from anther to stigma.
हाँ.
बराबर है कि नहीं? एंथर से पोलन ग्रेन्स निकलेंगे, वो जाके स्टिग्मा पे जाके डिपॉजिट होने,
हाँ.
उसमें से आगे की एक्टिविटी होगी और फिर बाद में अपना डबल फर्टिलाइजेशन चलता रहेगा.
हाँ.
समझ में आया आकांक्षा?
हाँ.
सी ऑटोगैमी मींस ऑन अ सेम फ्लावर, वहीं पे उधर ही
Same plant body.
बाजू वालों को भी नहीं देखना है उनको.
हाँ.
अंडरस्टूड? तो दूसरा प्लांट तो भूल ही जाओ.
हाँ.
गिटिनोगामी मींस सेम प्लांट बॉडी बट डिफरेंट डिफरेंट फ्लावर्स.
हाँ.
एक ही रोज प्लांट पे दूसरे दूसरे जो रोज है उनके बीच में ही अपने आपस में ही उनका पॉलिनेशन चल रहा है.
Okay.
अंडरस्टूड? दिस इज
हाँ.
कॉल्ड एज एन व्हाट? दिस इज कॉल्ड एज एन गिटिनोगामी एंड ऑटोगैमी.
Geitonogamy.
Self pollination, again I am repeating, self pollination means pathway is from anther to stigma, and how the pollination is happening from the same flower or another flower, लेकिन same species के
Same
plants होने चाहिए.
species.
Understood,
हाँ.
Akansha? Okay. Now, this is a pathway how the pollination activity has been carried out. All right. So till here
हाँ.
you have understood.
Yes.
Yes. Now the thing is, Akansha, today we have studied what? Today we have studied about the significance of double fertilization, pollen pistil interaction.
Okay.
Even we have studied development of endosperm. We have
Yes.
studied
Type.
about, if you will see, yes, we have studied the types of endosperm. We have studied regarding the, and we have started with the pollination types and even the agencies. Right?
Yes.
Okay, dear. I'll be giving you homework related the same topic only into the group. Right?
Okay.
You will be sending me homework till tomorrow evening.
Okay.
All right, I'll be giving you the
Yes.
related topic homework only itself. And Akansha, after finishing of the chapter, there will be a test.
Okay.
All right, I won't take a test
Yes.
of single topic. I'll take a test of lump sum of whole chapter.
Okay.
All right, till that time you start preparing because soon we will finish up with this chapter. All right, only
Okay.
few
Okay.
slides are remaining. Okay,
Okay.
from here the pollination agents which are there, that is entomophily, anemophily, hydrophily, or chiroptelophily, right? This all are very easy to understand and very
Okay.
very easy to remember also. I'll give you the tricks how to remember, because usually students get confused, right, during
Okay.
remembering. So I'll give you the tricks how to remember and what are the way to remember, the proper way to remember all these points, right?
Okay.
This will get over fast, and then we will be soon finishing with our chapter also, right? So
Yes.
you start preparing so that we can move ahead for the test,
Okay.
right? Any kind of in doubt till here, Akansha?
No, ma'am.
Till now you have referred the chapter?
Yes.
Yes. If you have referred the chapter, have you studied with the chapter properly, thoroughly?
Yes.
Any kind of in doubt you have found, that any kind of in word you are not getting it?
No.
Till now everything is clear.
Yes. Yes.
Today you have understood till double fertilization.
Yes.
Okay, fine. So this all are the terminologies. Expected questions. I have said you till types of endosperm. Regarding this pollination activity, dear, definitions are asked. All right, individual for a 1 marks. If the 3 marks question is there, so what they will do, they will ask you in an individual form, that is, define self-pollination, autogamy or zytogamy, right? Or they can ask you that what is pollination and explain the types of pollination, that is, explain the types of self pollination. All
Okay.
right, and this way the expected question can come into a board exams. All right,
Okay.
and in the next lecture which we are going to study about the pollination, remember dear, this that pollination patch is not only important according to the theoretical point of view, but even it is important according to the practical point of view.
Okay.
All right. So the points which I am going to show you in our next lecture, Akansha, it will help you during the theoretical papers also and practical also.
Okay.
All right. So same points you can carry forward into your practicals also. You can write it down into your practical examination also. All right, Akansha.
Yes.
Yes. Soon I'll be putting up the homework. Are you feasible with the diagrams?
Yes.
And the diagram which are present on the diagram of endosperm, dear, which are present on the page number 11. All right, so that page
Okay.
number 11, types of endosperm, figure 1.14, right? Those diagram you have to practice it regarding nuclear endosperm, cellular and halobial,
Okay.
because if it is asked individually also, you have to draw it. All right,
Okay.
and the diagram of double fertilization.
Okay.
All right, dear.
Yes.
Any kind of inquiries for today's lecture?
No, ma'am.
Okay, so I'll be soon putting up the homework also, dear. So today we are
Okay.
ending up with our session. All right, dear.
Yes.
Thank you for attending the lecture. Have a nice day ahead. Take care.
Yes. Bye, ma'am.
Bye, beta.